Hey everyone, and welcome to this explainer. Today, we're diving straight into the absolute cutting edge of immunology and genetic engineering. We're going to explore what it actually means to upgrade the biological engine of modern medicine. Since the massive success of mRNA vaccines, the whole world has basically woken up to the insane potential of RNA therapeutics. But, and this is wild, as incredible as that technology is, it's really just generation one. We're actually standing right on the brink of an entirely new era.
So I want you to start by thinking about this. What if a vaccine could literally multiply itself inside your body? Just imagine a world where we don't have to inject these massive, heavy-handed doses just to force your immune system to pay attention. What if instead, we could just drop off a tiny set of instructions and the vaccine does all the heavy lifting? It just autonomously prints more of itself once it's already safely inside your cells.
Well, to understand how we actually get there, we kind of need to look at how this tech is evolving. We all know standard non-replicating mRNA vaccines by now, right? They're super effective, but they require pretty high doses, usually somewhere between 30 to 100 micrograms. And because of that big dose, they stimulate your innate immune system pretty aggressively. That's what gives you those immediate, achy, feverish side effects, what scientists call reactogenicity. But then we have the upgrade, self-amplifying RNA, or saRNA. Because it multiplies autonomously inside your cells, a recently licensed saRNA vaccine only needs a remarkably low dose of just 5 micrograms. A much lower dose means way fewer of those harsh side effects, but you're still getting that incredibly durable immune response.
But here's where things get a bit tricky with the physical structure. Standard mRNA is relatively short and sweet, but self-amplifying RNA, that molecule is absolutely massive. And that's because, in order to self-amplify, it has to carry this incredibly long genetic sequence that codes for an amplifying enzyme, a replicase. It's essentially carrying this giant engine on its back. Having both the engine and the payload on the exact same molecule makes the RNA sequence so incredibly long, and that poses some huge real-world challenges for manufacturing, delivery, and even just keeping the vaccine stable. And honestly, that brings us to the ultimate plot twist, Trans-Amplifying RNA, or taRNA. Section 1. Splitting the engine, Trans-Amplifying RNA. Okay, so this is where the story shifts from just upgrading the vaccine to completely re-architecting the whole thing from the ground up. Scientists basically realized, hey, if the big problem with saRNA is its massive, bulky size, maybe we don't need to keep the whole system super glued together. By actually splitting the replication system into two entirely distinct parts, we get trans-amplifying RNA. I always like to think of this bipartite or two-part system using an engine and blueprint metaphor. With the older saRNA method, the replicase engine and the antigen blueprint are physically joined together as one giant piece. But with the taRNA mechanism, the system is totally split. One RNA molecule is just the engine. It's a non-replicating mRNA that only codes for the replicase protein, nothing else. This second molecule is this remarkably short RNA called the transreplicon. This is your blueprint. It has the code for the vaccine antigen, but crucially, it doesn't have the engine attached. But once both of these enter the cell, the translated replicase engine zeroes in on the transreplicon blueprint and just massively amplifies it in trans.
400. I really want that number to sink in for a second. In a recent proof-of-concept study, researchers found that to mount a truly powerful protective immune response using this two-part system, you need an incredibly tiny amount of the blueprint. In fact, you need 400 times less of the transreplicon compared to the replicase engine. The amount of actual vaccine antigen RNA being injected becomes practically negligible. It's wild.
Section 2. Lessons from alpha virus history. How viral accidents inspired a revolution.
Now, scientists didn't just magically invent the split engine concept out of thin air. They actually figured it out by studying natural viral accidents over the last 50 years, specifically in a family of pathogens called alpha viruses. You see, RNA viruses replicate incredibly fast, and their natural replicase engines, well, they don't have a spell check function, so they make sloppy copying errors. Sometimes these mistakes lead to massive chunks of the viral genome just getting deleted, which creates what scientists call defective interfering particles, or DIPs. These defective genomes completely lose their own engine. Now, on their own, they're totally harmless because they literally can't replicate. But if they happen to infect a cell that's already infected by a healthy helper virus, these little defective genomes act like parasites. They basically steal the healthy virus's replicase engine to rapidly multiply themselves, and they often end up completely outcompeting the original virus. Fast forward to the 1980s and 90s, and these brilliant researchers who were studying viruses like Sinbis and Samuliki forest had a total light bulb moment. They realized they could totally hijack this exact viral parasite mechanism. They figured out that if they engineered their very own defective particles, but swapped out the viral payload for foreign genes, they could just use the helper virus's engine to amplify whatever gene they wanted. This two-part gene delivery system laid the literal foundational groundwork for the modern tRNA technology we're talking about right now.
Section 3. Perfecting the vaccine recipe. Engineering the transreplicon.
Jumping back to the present day, today's scientists are using all that alphavirus history to optimize our modern transreplicon blueprint. Early versions of the transreplicon still had some pretty clunky leftover remnants of the original alphavirus replicase gene, plus this thing called a subgenomic promoter. But by meticulously trimming away all those unnecessary parts, researchers streamlined the blueprint down to its absolute essential core. They call this the shortened transreplicon, or STR. Practically speaking, it looks and acts just like the conventional mRNA vaccines we use today. But because it keeps these highly specific conserved sequence tags at its ends, it has absolute, undeniable replication superpowers.
So how does this all play out inside your cells? Well, there are 3 really elegant steps to tRNA vaccination. Step 1, the replicase RNA enters the cell and gets translated into engine proteins. And a quick side note here, this engine RNA does not copy itself. It literally just builds the engine. Okay, step 2, those new engine proteins scan the cellular environment and recognize those highly specific conserved sequence elements, or CSCs, that are basically capping the ends of our shortened transreplicon blueprint. And finally, step 3, the engine locks on and triggers this massive exponential transamplification of the blueprint, totally flooding the cell with the protective vaccine antigen.
Section 4, ready for future pandemics. The massive implications of tRNA.
Okay, so why does all this intricate molecular biology actually matter to you and me? It matters because this split engine design fundamentally changes our flexibility, our speed, and our safety when we're preparing for future pandemics. The real-world benefits here are staggering. First off, let's talk side effects. We learned from COVID-19 that if you modify RNA with a specific nucleotide called N1-methylpseudouridine, it dramatically lowers reactogenicity, basically way fewer side effects. But you actually can't put that modification into standard self-amplifying RNA because it straight up breaks the engine. However, with TASCus RNA, we can put those modified nucleotides entirely into the separate engine molecule, which makes up the vast majority of the vaccine dose. Second, multivalent vaccines. Because the engine honestly doesn't care what blueprint it's reading, we could mix multiple different transreplicons into one single shot to fight several diseases at once without massively jacking up the total dose. And third, rapid pandemic response. This brings us to the absolute kicker, the total mass of a future tRNA vaccine. Roughly 90% of it is just that universal replicase engine. Only about 10% is the customized transreplicon blueprint for whatever specific virus we're fighting. Do you realize what that means? It means we can manufacture and stockpile literal tons of the universal engine RNA right now, completely independent of knowing what the next virus is even going to be. When a new seasonal flu hits or a brand new pandemic emerges, we only have to manufacture that tiny 10% blueprint portion and just mix it with the engine we've already got sitting in the freezer. The sheer speed at which we could deploy a completely new, globally scaled vaccine would be entirely unprecedented.
So by basically looking to nature's own sloppy viral accidents, scientists have effectively split the engine. They've solved the massive hurdles of RNA size, stability, and those nasty side effects. We're moving from thinking of vaccines as just static instructions to vaccines as dynamic, highly optimized replication machines. Which leaves us with one final, incredibly exciting question to ponder. If our vaccines can now replicate, scale, and adapt on command, what disease are we going to eradicate next? Thank you so much for joining me for this explainer. I really hope you found this as fascinating as I did. Keep learning, and I'll catch you next time.